Good morning, everybody. I'd like to thank the organizers. It's been a lot of fun being here so far. And my name is Sayan Mukerjee. I'm in Duke. I'm in mainly math and stats and a few other departments. But what I mainly do is a lot of methods development. And what I'm going to talk about today is modeling microbial time series. And what I'm going to focus on is thinking about compositional data, and I'll define that and talk about why. Louder?
We can't hear you, sir.
You don't hear me? Okay. Is this better? Okay, great. All right. I'm going to talk about why we think of this as compositional data and some of the challenges that are involved in this. And I'd say this talk is somewhere between a tutorial and a research talk, and I'm going to try to touch on some of the ideas on Monday that Lior and Sharon had talked about. And a lot of these, kind of this perspective or presentation is a lot of work that I thought about with a student working with me, Justin Silverman, and my colleague, Lawrence David, who's does some really cool experimental work. So we'll start. Microbiome, what and why? So what we think about in terms of microbiota or microbial data is a collection of microbes, and you can go look and see that there are a lot more microbe cells in your body than you, and so on and so forth. And when we start talking about the microbiome, what we mean is the collection of microbial genes associated to a host. And what we will measure using the technologies that we often look at is the microbiome, and often we reinterpret that as microbial data. Okay? So people have been thinking about microbial data for a while. This is from 1929. They took some Arctic explorers and gave them meat for a year and saw what happened to them, and sometimes not so good things, but you know. So people have been very interested in this for a long time, and as you see, this has been going on. So what's really changed is sequencing, right? So with sequencing, you can get a lot, and you can look at a really wide diversity of microbial communities. And again, this is just an example here. So if you have to culture things, right, you have about 40% species you can't get to. Sequencing, it goes down a lot. The number of bacteria that you can identify is just increasing dramatically with sequencing technologies, as well as the time to results. Now, I'll come back to this at the end. This 2 days, it sounds good, but as we go forward, we do want to get things faster. Okay. I'm just going to spend a few slides talking about why people have been thinking about it and just some properties of microbial data. So there's a wide diversity between different types of body sites in terms of what microbes are there. But what I think is even more important is the diversity of the microbial data at different body sites. And this plays a part in a variety of issues. This was a very interesting study where you can make skinny mouse fat. You take a skinny mouse and if you take these fat mice and you give them basically a fecal transplant, take their microbiome, stick it into a skinny mouse, you can make a skinny mouse fat, and vice versa. Now, these are all nude mice, so they don't really have an immune system, so that affects things a little bit, but this was a very striking example. And one place where really in medicine you're seeing a lot of use and people thinking about the microbiome is for Clostridium difficile, or C. diff. And one of the things people notice, if you get certain antibiotics, you go to the hospital, you get certain antibiotics, it's almost like a wildfire, right, in a forest. It wipes out a lot of your microbial community. Some of the ones that live and stay and dominate are quite bad. And how you treat this, a really common way that people are treating this now is by a fecal transplant, and that you take possibly your microbiome previous to surgery, and then you come back and you stick it in after surgery, and this is a really common treatment. Now, this idea of fecal transplants, it's interesting. At the Duke Lemur Center, they've been doing it for actually 20, 30 years. So this is a sifaka. It's quite cute. It's a lemur. And it was sick, and this is one of the ones they gave a fecal transplant to, and that's just a picture of its gut, or supposedly what its gut looks like. Okay, so this is just some general background as to why people are thinking about this and why they care about it, and there's a ton more which I won't go into. But there's some really basic questions, right? You know, if you watch TV, you see all these ads for probiotics. Eat my yogurt, eat this, eat that, it'll cure you of X, Y, Z, right? And you know, it's a quick question is, does any of this work? Does it, you know, what does it do? So, you know, what role does the microbiome play in disease? Can we design therapies in probiotics? And can it be used as a diagnostic or prognostic indicator of disease? So these are some really basic things people have been thinking about and want to get a better understanding of. And so my colleague at Duke, Lawrence David, has made a fake gut. So this is a chamber, right? And you can control the pH in it, you can control the temperature, and using bead technologies, you can actually inject probiotics in there. Okay, so you have some distribution of microbial community, you inject probiotics, and then you run a time series, and you ask, how quickly does the composition change? Is most of the temporal variation technical? Is it biological? What does this temporal evolution look like? And Justin decided to volunteer himself and took a lot of daily samples and hourly samples and replicate samples, and he had a lot of fun. So this generates a time series, and I'm going to talk about how to analyze this time series, but that'll be at the very end. Before we get to that, I wanted to just talk about all the stats and all the other things that go into this. So if you think about modeling something, the first thing you should ever think about is a data generating process. How is the data generated, and what does it look like? And so in this case, these are the steps. You have sample collection and storage. Could be, you know, one case I work with colleagues who study baboons, so it could be picking up baboon poop in Kenya. It could be, you know, various other things, right? But you need to worry about that. Then you think about the data extraction, and then the sequencing, and then from the sequencing, specifically all the data I'll be talking about is a particular type of data today. It's called 16S. It's a particular technology. From sequencing, you assign the sequences to samples, you denoise or cluster the reads, and then you get a bunch of counts, okay? So what I'm going to focus on today are, I'm not going to talk about this part, and I'm not going to talk about this part. I'm going to assume that these are kind of taken care of algorithmically. If you want to watch, I think videos, I think David has actually talked about those two parts previously. So I'm going to talk about what's going on in the top three. Okay, so I think this is what I just told you, okay? And you worry about counting in bias, technical variation in bias, and what's the actual biological variation in signal. Okay, so this is what we're trying to get to, and we're trying to understand. And so at the end of the day, these are the data that you get, right? You just get these different counts across these different samples, and then you have all these different species, and they're counts. We've been modeling count data for a long time, and we'll come back to that. Now, there's a really key point here, and this is where I'm going to start talking about compositional data, and this is really important for your thinking about the data generating process. And this also relates to, I think, the first talk yesterday about RNA-seq. There really are cases where sequencing depth does tell you something about the total number of counts, right? Probably bulk RNA sequencing is one of those cases. There are also cases where it really doesn't tell you much. And for various reasons, which we can get into later, the microbial case is one where it's really hard to kind of infer or correlate the amount of microbes you have to the sequencing depth, again, because of the technology, which means it's very hard to get an absolute count, okay? And that's one of the reasons why we're going to think about the data as compositional, and that I don't really believe the total numbers. All I believe are the relative frequencies. But you should really think about this as a knob. And there are various problems where I kind of believe the absolute numbers, but they're uncertain. There are other cases where I really do believe the absolute numbers, right? And you can think about this as a knob, and I'll talk to you later on as I go through some of these models as to where you can kind of turn that knob, okay? The other thing that I'll say, part of the microbial data, it's sparse. If you look at it, there are a lot of zeros. It is what it is, right? These are the data you get, and you want to model it. Okay. So if you think about things being compositional, it ends up you're living on a simplex, and I'm going to talk about modeling on the simplex and why it's a little bit hard. Okay. So this is my simplex. all my data are positive, and they add to a constant number k. You make it 1, these are probability densities, but you don't have to make them 1, but so you have that. If you want to make it a little bit more formal mathematically, that's your simplex. It's d-dimensional, okay? I have a question. How many degrees of freedom does this have, even though it's d-dimensional? D minus 1, right? Good. Okay, great. So there's some really nice things about the multivariate normal. I just want to interject, which aren't true for modeling on the simplex, which is there's a notion that lets you relate statistical independence with orthonormal or orthogonal bases. I'll show you a picture of that in a second. And that really, that relationship underlies what Lior talked about with respect to PCA and CCA. It's really that relation between the probability distribution and the geometry that's absolutely vital. The other thing is there's this beautiful linearity in the multivariate normal. If you take a multivariate normal, multiply it by, set it by AX plus C, right? You get a nice normal. And there are things you can do with that. So here, this is just a basic picture. So what are we saying? I have 2 coordinates, feature 1 and feature 2, right? What does PCA do? It tries to give me one direction, which explains most of the variance, and then direction orthogonal to it, which explains the second most amount of the variance. And these two, in a way, are independent, right? So that's a basic idea. So if these were my 2 original vectors, once I've rotated it, I get this principal component and this principal component. And you can just break down these projections. Now, there's something that's really important here. There's a notion of orthogonality, right? You have these perpendiculars. That's really important. The other thing that's really important are these 2 directions are independent. That's great. You get that in this multivariate normal setting. Another thing that's really nice in the multivariate normal setting is pretend that these weren't compositional, right? Let's say I just have two OTUs, 2 species, and then I move one. Treatment moves the gray to the dark, right? You can see the difference. It's like by -3. This has a nice geometry. I can look at it. I can understand this. This is good. What happens if we do this on the simplex? Now, if things are living in a simplex, right, they all have to add up to some number. Arbitrarily, this is 1, so they live in this little triangle. And so what am I going to do? Let's say I move the bacteroides up, right? That's my probiotic. I'm kicking in more bacteroides. Okay, I just did that. That's really exciting. Well, because things add up to a constant, the lactobacillus has to go down, and the ruminococcus has to go down, right? So just by this construction, by the constraint that everything has to add up to 1, if you increase something, the other 2 have to go down, right? That's life. That's all you have. That's not like this case in the linear transformation, right? You don't have a nice coordinate system. You don't know how to think about this. And this creates negative biases and spurious correlations. So let's say there are 3 categories, right? Water, I don't know, last one's water. You have mineral, whatever. 2 other categories make them up. Animal, plant, I don't know. And so you take your object and then you desiccate it. You just dry out the water, right? You just take your hair dryer and you go to town on it, right? You're going to lose one of the elements, right? So the thing's going to get renormalized. Now, if I look at the correlation between sample A and sample B, you're going to get false correlations, both positive and negative. You have to, right? Now, is that a false correlation or not? I mean, it depends on how you think about things, but axiomatically, if you believe, right, that studies performed on subcompositions should not stand in contradiction to those performed on the full composition, right, then that is a spurious correlation, okay? You probably also want permutation invariance. If I, scramble the ordering of the parts, my analysis should be the same, right? And you want scale invariance. Whatever that constant was, if it was 5, 10, 3, it shouldn't matter, right? So these are just some basic invariances that your analysis should probably satisfy if you are really believing that it's compositional data. Okay, so what I'm going to talk about for a little bit now are how do I get back to something which looks more like the normal world, right? For these types of compositional data. How do I transform them in a way that lets me do what I could do with a multivariate normal, okay? So there's this idea of this log ratio transform, okay? So the first thing is it's called the CLR, which is LR stands for log ratio, C stands for centered, and what I'm going to do is, actually let me just write it down here. I have x1 up to xd, right? This is my set of compositions, okay? And what I'm going to do is I'm going to transform them, and I'll tell you why I'm transforming them in a little bit, but first let's just transform them. And so what I'm going to do is I have a normalizing constant, let's call it, for lack of a better term, let's call it kappa. And my normalizing constant is just a product over I, xi, and let's say I is equal to 1 to d, and this is 1 over d. Okay? One of the things that you're going to be noticing is that normally when we think about the multivariate world, we think about adding and multiplying, right? You often end up adding a lot of stuff. Here in this compositional world, you see a lot of products and you see a lot of logs. So you add logs and you have products, right? So the operations multiplication becomes like addition. Okay, so now I have this kappa, this is my normalizing constant, and so each coordinate, let's say I'm going to transform x1, let's call it y1. This is going to transform into y1 up through yd. Okay, and what we're going to say is yi is going to equal the log of xi over this normalizing constant kappa. Okay, so basically I've just log transformed my data, which is something that we really often do. We often are log transforming our data and analyzing those as a normal, right? But here we're doing a little bit more. We have these these normalization factors, okay? Now, the second thing I'm going to want to do is I'm going to want to define some notion of orthogonality. So, as I'm telling you before, right, I'm living in a simplex, okay? So now I want to tell you what does orthogonal or what does orthonormal mean in a simplex, okay? So I have these compositions. Let's say it's a collection E1 up through ED minus 1. Now, the reason why I have d minus 1 bases, right, and not d is because, again, we know the last one, right, because it sums to a constant. Okay, so far so good. Okay, so now what I'm going to say is that any E1, okay, here is going to be orthogonal to, let's say, to E2, let's say EI to EJ, right? If I do this transform, right? If I do the CLR transform on them, okay, that gives me a new vector, right? So let's say we have CLR of EI, okay? And then I just transpose that, multiply that by CLR EJ, right? If that's equal to 0, right? These are basically orthogonal. So it's a standard idea of what orthogonal or perpendicular mean, but it's on this transform space, okay? Okay, so far so good. Now, there's another thing that we need to do, which is there's something called a basis contrast matrix, and I'll show you why we have that in a little bit. But it's a matrix. It's d minus 1 by d. And all I'm doing is taking these orthogonal elements, right, and looking at their CLRs and just stacking them, okay? Why I have this, I haven't told you. I'll tell you in one slide, right? And how I actually find these orthogonal bases, I haven't told you yet. I'll tell you in 3 slides. But yeah.
This is a linear space.
This is a really good question, okay? So if someone gives you a transform and says it's a good transform, the first thing you should ask them is this a linear vector space, right? And if they tell you no, then you're like, you're kind of shady, right? And the answer is yes. This will be a linear vector space. There's one caveat, which I'll get to, which is a big caveat, which is called 0, right? But I'll get to that. But that otherwise, yes. Okay. All right. So basically, what I'm taking is if I could find these orthonormal bases, right, and transform, and by definition you define them as orthonormal according to the transform, and I stack them, that's called a basis contrast matrix. Okay, great. So this is a key slide. The key slide in this whole transformation approach is that you have something called the ILR, which is called the isometric log ratio transform, and it has an inverse. This is a one-to-one map. So if I go, I come back, I end up in the same place, okay? And what is it? Well, let's just write a few things down, and then I'll come back to it. Okay, so first thing just to remember is okay, what did I do? I first told you previously what this, on that side, this is my CLR, okay? Everyone just remember, that's the CLR. Now the other thing that I told you, I have this contrast matrix psi, right? Which is just a stacking of the CLR, LR of E1 down to the CLR of ED minus 1, okay? Okay, so just remember that we have these two. Okay, now there are two transforms. Well, maybe I can just do it here. What I can look at is the ILR takes a CLR, okay, of X and multiplies it by the transpose of this basis contrast, okay? So if we think about this, this is going to take something that's d-dimensional and take it down to something that's d minus 1 dimensional, right? And you're not losing any information because that original d-dimensional object was really d minus 1 dimensional because it had added up to a constant, right? So that's the first thing, okay? Now, so that's your transform. Now there's an inverse transform, and what the inverse transform is doing is you have an object y. Y is actually rd minus 1. So this gets back to the question, y is actually in our standard Euclidean vector space, right? So what you can do is once you're in y, you can go back to living your life in multivariate normal land. And we're going to be doing that in a little bit, right? But that's why I'm introducing this, okay? So y is in our happy, I'm normal, things are Euclidean, life is good space. So now what I'm going to do, I multiply y by psi, okay? So this is going to take a d minus 1 dimensional object and turn it into a, anyone? D-dimensional object, right? So which is good, okay? And so then I exponentiate it, which maybe is not shocking because how do you unlog a log? You exponentiate, right? So so that's fine. This is going to be a vector. Let's call this, I don't know, y tilde 1 up through y tilde d, right? These are all going to be positive by construction. And then I just renormalize it. So now I get yi is just going to be y tilde I divided by the sum over, let's call it j, y tilde j, where j is 1 through d, right? Okay, so that's how you come back from this, oh, I should probably leave this here. It's early. Okay, so that's how you come back and do the inverse transform. Okay. Now the question is, how do you get the basis, right? I haven't told you yet how to construct psi. Okay, so it turns out that that if you have a phylogenetic tree, you can construct psis. And I'll tell you why, okay? So one way of thinking about a phylogenetic tree is partitioning. It's a recursive partitioning, right, of your coordinates. Okay, that's a very natural way of thinking about it. And actually, let me write it on the board, okay? And let's, for a little bit, pretend I had a phylogeny, right? I have these taxa, and let's say someone actually gave me a phylogeny of the taxa, because no, they maybe probably actually did, okay? So now I'm going to try to build a matrix, let's call it theta, okay? Where theta ij is just going to have the following property, okay? What I'm looking at is I is indexing the nodes, okay? And what is theta, and what's theta ij telling me? It's telling me that node, do I fall into, so let's say I'm looking at node 1, right? So taxa 1 falls into one partition, taxa 2 and 3 will fall into another partition. So one gets a +1, the other two get -1s, okay? This is called a sign matrix, okay? At each partition, you may not be discriminating or separating all the taxa, right? So if we went to node 2, taxa 1 is completely ignored, and taxa 2 and 3 are being partitioned, right? So if we looked at node 2, taxa 1's ignored, so it's a 0, and then you get a +1 for taxa 2 and a -1 for taxa 3. Okay, so this is my signed matrix. Okay? Great. Now, if you sit down, do some math, or read math that people have done, you can realize that you can, given the signed matrix, you can construct these orthonormal basis contrasts. Okay? And the idea is you look at your theta matrix, and if you say, hey, look, if theta ij is equal to 1, then what I'm going to do is ni plus is a number of +1s in row I, ni minus is a number of -1s in row I, okay? And you use that formula. And this gives you this basis contrast matrix, okay? Now, specifically, the reason when we worked on this and we called it PH-ILR in our paper is we said we were doing a phylogenetic ILR transform, right? So when I talked initially about this transform, and I told you about this basis contrast matrix psi, right? It had nothing to do with phylogeny. In general, it doesn't have to do it. You don't need it to have anything to do with phylogeny. You just need an orthonormal basis. You can construct it however you want. It just so happens that if you know a phylogeny, you can get it. Okay, great. So this is what starts being cool about this space. These are isoclines. So if I look at the simplex, if you're modeling text, something similar will happen, right? If you look at what isoclines are, so this is the highest probability region of this cluster, and then as I go out and I go out, right, these are what the isoclines look like, okay? Now, if you look at what's happening really near zeros, so this is a real problem whenever you're working with compositional data or modeling, actually even things that are multinomial, is right near the boundary of zeros, the change in the variance is crazy fast, okay? So zeros are a serious problem. Now if I look at the middle, it starts looking like Euclidean space again, okay? Now what's kind of cool is once I've done this coordinate transformation, what have I gotten back? Right? That looks normal, right? That's kind of the beauty of this. It looks normal, and I can start using all of my standard tools that I have, in my, or someone's statistics toolbox. This is another thing that's cool. Let's say I have exponential growth trajectory over time. I have exponential growth, okay? So this one's growing exponentially, this purple's growing exponentially, and I'm showing you what it looks like in the simplex. If you map this back to this coordinate representation, you get straight lines that are parallel. Okay, and the direction is whether it's positive or negative growth. So this is another thing that's kind of cool about this transformation. So kind of as a summary of why we thought about these types of and it's not just us, why a lot of people have thought about various types of transformations for compositional data, is that if I start off with this type of community, right, this is my picture, if it's unnormalized, these are just my absolute abundances, and I'm just showing you how they move, right? So that's, in this space going from the red to the blue, you're moving in this direction, okay? This is what it looks like in terms of the nodes. My coordinates, if you think about this orthonormal representation, are not in terms of the species or the taxa. They're in terms of these nodes, right, which are the phylogenetic, they're called balances, right? And so you're kind of really looking at a log ratio of the bacteroides versus these two, or in this case, right? So this is what my orthonormal coordinate system looks like. It's really, one bacteria followed by a bunch of nodes, okay? So just something to think about and remember. So if I'm moving in this direction in the simplex, once I've transformed it, I'm just moving along one coordinate system, right? So this becomes a lot more easy also to interpret in terms of how you think about this in terms of a coordinate system. Now, there's one last thing that I want to tell you. A lot of my colleagues who model, okay, let's say someone decided to torture you and made you take a categorical data class. Has anyone taken a categorical data analysis class? Yeah, well, sometimes they're really good. One of my colleagues, Lima, teaches a wonderful, wonderful class. Sometimes they're really terrible, okay? And you just like, it's like, today we're gonna look at zeros and ones. Tomorrow we're gonna look at something that's Poisson-like. You know, day 4, I don't know, right? Anyways, but the point being is that something you see a lot is y is a multinomial vector. P are probabilities of each of these things, right? And you multiply this pi by something called a logistic normal. Okay? I'll show you the logistic normal in a second, but it's not fun. Okay? An alternative, really reasonable alternative, is you still have the multinomial. I do this ILR transform on pi to get an eta variable, the inverse ILR transform, I'm sorry, and that I can model as a multivariate normal. So I just stick this deterministic step in there. And there's a reason why I'm showing this, which you'll see in a second. This is a logistic normal distribution. Okay, you log things. Okay, so what does this mean? I take the first to the d-1 coordinates of y. That's a vector. I divide it by the dth one. I log it. I subtract off mu. This is my covariance, right? So the rest of this looks normal-like, which is, this is not terrible. But then in front of it, I have to multiply by this pi sub I, yi, right? And one of the things that I think Sharon mentioned is normalization for distributions are important. And if you have a normalization like this, where the random variable sits there, it can make life hard. And this is not an easy distribution to work with in general, or often. Okay. I told you I'd talk about time series, so let's end this talk with time series. Okay. So let's go through this slide a little bit carefully. All right. How many people here have heard of a, oh, my marker. Oh, we can use it, well, I'll use it. How many people here have heard of a Kalman filter? Okay, we're gonna basically be using a Kalman filter. So let me explain to you what I mean by that, okay? I have some parameter theta, which corresponds to the microbial states. Okay, it's not gonna be exactly the microbial states because that's something I observe, right? What it's gonna be is some ILR transforms of those probabilities, right? That's my hidden state variable, and it gets updated. Okay? What do you think is the most natural or easiest way of thinking of updating from theta t to theta t plus 1? I take some matrix A of the right dimension, I multiply it by theta t, right? I add some noise, let's call it nu, right? And I just call this theta t plus 1. Okay? Everyone's okay with that? Now I told you, hey, look, this isn't my real thing. Theta isn't my real thing, right? So what I'm going to do is there's actually something I observe. Let's call it y, because I called it y before. And I tell you yt plus 1 is equal to some b times theta of t. Again, let's say plus some noise wt. Okay? What I've written just out right now, this is a Kalman filter. This is called a Kalman filter. It's a very basic time series, also control system model. And one of the things we're going to start saying is, well, maybe these A's can vary with time, right? Just to make this more complicated, and maybe because it's true, okay? This starts looking like something called a dynamic linear model, and we'll talk about that in a second. But that's all we're doing here. We're just saying we have these thetas. They're being updated. This is somehow the true state of your microbiome. Into each of them, I inject some noise, wi. Oh, shit. I'm a terrible person. WT, VT, okay? So I inject these W's in there, right? And then you have an observation process, which goes from the thetas to the V's, okay? And this is just what I'm showing you here. And there's a really important thing, if you do biological modeling, if you can, you want to understand how biological noise is different than technical noise. And our W, we're going to be really thinking about as biological noise, and V's as technical noise. Okay. Oh yeah, and then you have your observed counts. Okay, that's our model. And here I just wrote it out. And this right here is called a dynamic linear model. If these F's and G's did not have T's, this would be a straight common filter. Okay? But these are variable. Okay. So we have a multinomial logistic normal dynamic linear model, which is called Mallard. And well, you can see why the duck is there. It's Mallard duck. And there's a really important idea that Justin came up with, which is every once in a while, instead of just doing longitudinal sampling, you need to do replicate sampling. And the reason why we need to do that replicate sampling is to be able to get an estimate of the technical variation. If we did not do that replicate sampling, we could not get an estimate of the replicate variation. Okay? And basically, our replicate samples lets us see what V looks like, what this uncertainty and distribution in V looks like. Okay. Okay, great. And just, this is how we deal with them, but that's technical. Okay, so Justin simulated data, and this is what it looks like. And then what we're showing is our posterior estimates for theta with the 95% credible interval. And this is a pseudo count estimate. Purple is a true value. It's kind of hard to see in here, but the point being is that we actually did okay, is all we're trying to say. Because the blue and you can't really see the blue, can you? I guess that's a good thing, because it's overlapping with the green. If you were on my screen, you could see it, but really not that great either. But the point is, they actually do overlap reasonably well, which they had to, because otherwise I would not be sitting here and telling you about it, right? If it doesn't work on simulated data, maybe there's an issue. Anyways, but what's also interesting is you start getting an idea for what these uncertainty looks like, right? These are my posterior estimates. Green is a true value. Now you can start seeing the green, and you can start looking at what do the W's look like versus the V's, right? So you're saying, what is the difference in the variance that's coming from technical versus biological, right? And so that's kind of interesting. And again, this is just to show you an example with 3 of them, okay? Okay. So now if you think in terms of variation, right, you can actually construct this matrix of these log variations. Okay, you can think of basically the analog of a covariance matrix. You can start trying to think about how these various communities are covariating with each other, right? And you can look at how that's looking either in terms of technical variation or biological variation. And then you see some weird things happening, and you can go back and say, oh, there's a feed disruption, but that's a technical thing. Some of the things that Justin found, there are sub-Daly dynamics in this chamber. And so this is an important point. Time series modeling is computationally painful. It's costly. There's a bunch of stuff. I didn't tell you how we did inference because I hid that. It's a little bit ugly. The actual inference algorithms to do this. We used something called INLA, but it's a little bit hard. And without simplifying assumptions right now, you can do about 10 bacterial families, 700 samples. Takes about 2 hours. If you simplify it, it's about 10 minutes. Approximate inference, you can do 1 minute, but you start losing things, right? So thinking about inference here is really important, and if someone wants to think about inference and scaling these problems, I would be more than happy to listen to you or to talk to you about it, okay? So these are some of the challenges. Take-home points. Depending on what your sequence data are and what the generating process is, it's, okay, let's just say always. In some sense, it's always relative information with count uncertainty. If the count uncertainty is 0, right, it's no longer relative at all, right? You know exactly how many there are, but often there could be a ton of count uncertainty, and especially in the microbiome, just the way microbiome data are generated, it's hard, right? If you're modeling something in general, if you can try to get some estimate of the technical variation, it's a good thing to do. And the last thing is, these dynamic linear models, there's a reason we chose them. People have developed them. They've worked on them a lot. They've been used extensively in things like finance and a lot of engineering applications. So they are reasonably flexible. So that's one of the reasons why we also used it. And I think that's it. If anyone wants, well, I have another project with people where we used variations of dynamic linear models to generate music pieces. And if you go to my website, you can see one. I can play one for a second, but let me stop and first take questions.
Any questions? Yeah. You said that part of the motivation for taking the ILR is to get an orthonormal basis for things that are normal, right? Is that still true when you have overinflation of 0?
Right. Okay, so let me repeat the question, right? So the question was that I have this ILR and we used it to get an orthonormal basis, right? And I guess Tyler's question was, what about overinflation of zeros? Okay, so one of the real problems in count data is, and this is called zero inflation, is that the most natural way of thinking about count data is a Poisson distribution, right? Everyone says, hey, look, you've got counts Poisson distribution, okay? Now, if you look at a Poisson distribution, and if you look at your data, and there's a massive pileup of zeros, the Poisson does not fit that well, right? So one of the early cases where people actually were looking at count data were epidemiologists, who I think were one of the examples is cholera in Wales in India, and they had this big overinflation of zeros, okay? So there's a really basic problem in thinking about these ILR transforms, which is zeros. Dividing by 0 is a problem, okay? So zeros are a serious, serious problem. So I don't know what to do with the 0 in a way. What I can do is I can take those zeros and I can put them somewhere else, right? Somehow model with them with something else, okay? And then model kind of the remaining things with the ILR, right? So what you can do is you can think about a mixture model where you have some things modeled by a delta function basically at 0, and then you have the rest. And for the things with the delta function, we're going on and doing all of this machinery. I mean, for the non-delta function assignment ones, you're doing all this machinery. For the deltas, you just work with that delta, right? That's one way with dealing with it. If you do deal with it that way, computationally becomes a lot harder because you have to have this latent variable, which is assigning to that sample whether it's, belongs to one or the other. So I don't know if that answered your question. I guess the answer is zeros are hard to deal with the ILR transform.
So in the first work that you presented, you also in the paper offered a way to weight the zeros.
Yes.
Can that be a solution to that?
So yeah, so there's a solution to that. You can think about weighting the zeros in different ways. So let me say it this way. There are a lot of ways one can deal with zeros, okay? We're actually, Justin and I are writing a paper right now called Not All Zeros Are the Same. And the reason why I'm hemming and hawing a lot is that not all zeros are the same. And depending on what the biological process is that's generating the zero, you might want to think about it in different ways. So one way you can get a zero is you just missed a measurement, right? Another way you can get a zero is there's nothing there. Another way you can get a zero is there really something there, but it was too low, right? It was just in that, not in the, your machinery can't measure. And depending on all of these things, you might want to do different things, right? So this is why I'm hemming and hawing a little bit.
One more question. Very cool.
Yeah. For the fake gut model, you guys use like a bioreactor or a robot?
It is, this is something that they made. So it is really a bioreactor. And if you, it is actually what I showed you. And it's a little bit smelly. So it is actually just this thing that sits in the corner of the lab, which has these, I don't know, these glass things in them. You can see I don't do experiments, right? But you have these glass things in them, right? And you have all of these tubes, and you can actually control the temperature, and that's why you have all of these pipes going in there. And then you have microfluidics hooked up to it, and you have these little, so yeah, this isn't the only one out there, right? There are a variety of these types of reactors that are there. And yeah, these things are also shaken, and yeah, because you don't want the shit to hit the bottom. Anyway, so yeah.
Excellent. Well, let's thank the speaker again.